Hello. Ooh, let me turn the lights off. This is almost emotional. This is like the last one of these PowerPoint based kind of knowledge things. We'll do a little more practice after this, but we're getting down to the end. So, this is a cute one. You like this, you gotta like this. So, here we go, right, with genetic engineering. They have taken out about everything about this. I'm going to start with this, right? Beyond the scope of the course, details of these processes. These processes are basically three, and they're telling you don't need to know how they work, really. The focus should be on the application of these techniques, conceptual understanding. I honestly can't imagine that they'll ask you anything here except dogma related stuff, because that's again what we have, and we'll get back to that. But here I put explain the use in analyzing or manipulating DNA. The illustrative examples they said teachers can use in class talk about amplifying DNA fragments, propagating. That means the same thing. It's a little bit redundant, right? But two different methods of which we're going to talk about. That's two of their three techniques they talk about here. Um, Analyzing DNA, that's like pretty much the fourth technique or the third technique. And you can use that to identify a criminal or identify an organism, which a criminal is an organism. So there's some more redundancy, right? This phylogenetic analysis, that's evolution stuff. They're not going to ask that. And then they're saying that if you modify an organism, that can also include animals, but they don't then say like what you might do with them. So very vague. This should be pretty quick. Here we go. Okay. Transformation is one of those processes that they just mention specifically here, but say just need to know like that you can use it. But transformation was something that we talked about before. What did we talk about transformation before? As a method of recombination in prokaryotic cells. What did that all have to do? I can give bacteria cells a new trait, which is due to their ability to make a protein, which is due to their having some RNA, which is due to the fact that they got some DNA just like by soaking it up, which is what transformation is. And when we humans learned that bacteria cells can do this, we started experimenting and figured out it was actually very easy to get a human gene into one of those plasmids, which is what soaks into a prokaryotic cell, and then have it soak into that prokaryotic cell, soak into, transform, right? And then that bacteria cell has new abilities. Maybe it can make some kind of medicine, but they didn't put that on their list in any way, shape, or form. But to make a new protein with the use of transformation, they might work that in a story somehow, but that story always revolves back to the dogma, because making that new protein is a trait that that thing never had, and you gave it to it by this method of getting DNA in it. But what it can also do is make copies, clones of that gene that you got in there, because every time it does that binary fission thing, these prokaryotic cells copy all their DNA, including that plasmid with the gene you got in there. So if you want to get that gene back, you just have to take it back in a similar way to the way you got it in there, which involved cutting with restriction enzymes, which is nowhere in the syllabus. So we talked about this, and all that stuff we talked about is nowhere need to know. So I'm skipping it. Okay, so gene cloning was one of the things, and transformation to make clone genes was one of the things they had in the blue box. Another thing they have in the blue box is PCR. Here's how DNA fragments are amplified, right? But here's what, we emphasized this before, as of now, this PCR method can only copy small pieces, and genes are not small pieces. So if you want to make copies of whole genes, you still have to do it the cloning way, which involves transformation and letting bacteria cells actually make it for you. But if all you need is little pieces to analyze and identify, like criminals or, again, evolution stuff, then this can make small pieces without cells in a test tube kind of, or in this fancy expensive machine by doing this, which you don't need to know how it works. But it's all about complementary bases and stuff like that. And you're basically in this machine replicating replication. So after you've cut DNA and amplified it, likely using PCR, right? The fragments now you can separate by gel electrophoresis, and this made the blue box. This is the third technique, transformation, PCR, electrophoresis. It's going to separate any kind of molecules, really, but here we're mostly talking about DNA, according to size and charge, and that's all they say. Remember, they told you you don't need to know how it's done. DNA sequencing can be done using these little barcode patterns that can also help you, again, identify a criminal or children, parents of children, stuff like that we talked about. But none of those really made the blue box list, right? But by determining the order of nucleotides in DNA, you can identify one kind of organism compared to another, and maybe if an organism has this particular gene, because that's all about the sequence of bases in a DNA. So you got that could be tied in. So now transgenic organisms, this is I think our last one. This again did not make the blue box, but it made a genetically modified organisms with no requirements to know how they were modified, and that's a difficult thing. To get a gene into a eukaryotic thing is much harder. Remember what we said before, viruses would be the vector of choice, but they didn't have any indication that they would ask you any of that in this case. So these sheep are in the picture because they were modified, given a gene that allowed them to make this protein, which can help hemophiliacs. And so to make a medicine this way in genetic engineering all has to do with what, again? The central dogma, right? Why did these little creatures have this new trait of being able to make this protein? Because they got some DNA that made RNA. This is getting messy again. That made this protein that gave them this new trait. So again, all of this also could be traced back to the dogma. I don't think they'll ask you anything else. So anyway, Duck walks into this bar. The duck says, "You have any grapes?" Bartender says, "This is a bar. We got beer. We don't have any grapes." Duck comes back the next day. Says, "You have any grapes?" Bartender says, "Look, I told you yesterday. We don't have any grapes. Now, if you keep bugging me, if you ask me that again, I'm going to nail your little duck bill right to this bar." Duck walks out. Comes back the next day. He says, "You have any nails?" Bartender says, "No." Duck says, "Well, do you have any grapes?" That might be the best one.